You already know that I think that proteins should be considered amazing machines. Yes, they're made out of they are biomolecules, and yet they're not exactly inanimate, right? You saw them moving in the animation. Anyway, when we look at different proteins like this one, we can almost predict what they do. One of your labs, you may have already had it, is about a special group of proteins that are known as enzymes. And enzymes are the proteins that are most like a machine. Enzymes are almost always proteins. If your lab instructor says they are always proteins, I'm not going to disagree with that. There is a technical exception, and that has to do with the ribosome. But the rest of the enzymes, they are proteins. And proteins act as what are called organic catalysts. Catalysts, let me write that for you. Catalyst, Catalyst. Catalyst. Catalysts are substances that will make a chemical reaction happen faster, always faster. Catalysts never make things happen more slowly. Now, chemists, long ago when they were trying to turn lead into gold or whatever, they discovered these things called catalysts, and they used them all the time. Then one day, someone discovered that there were biomolecules that acted as catalysts, and that was a big discovery. And those catalysts are called enzymes, and they're almost always proteins. Now, enzymes, remember, they're like little tiny machines, I think of them as little tiny robots, and they will either grab one thing and rip it apart. And once they rip it apart, they let go of the pieces, or they'll grab two things and smash them together. And once they're together, they let go of the pieces. Or sometimes they'll grab a couple of things and rearrange them, but that's getting kind of fancy then. So one of the things that you should know or just memorize, and we will talk about it later, is that there are substrates in chemical reactions, and then there are enzymes in chemical reactions. And finally, there are products in chemical reactions. So let's talk about that, okay? So substrates. Substrates are what you start out with. The substrate or substrates is what's there at the beginning. And the product or products is what's there at the end, okay? So you start with substrate, you end up with products, before and after. Now, this arrow indicates that what happens between the before and the after, and it's usually left as a magical time, right? Stuff happens. Sometimes the name of an enzyme will be written right here above the arrow, but not always. Here's what's important. Let's look at this image. An enzyme, it's depicted as this green thing here. An enzyme has got a region called the active site. The active site. Now, since I think of enzymes as being like robots, their active site is like their little grabby hands. The active site is where, and you might want to write this down, where that protein called the enzyme will form a temporary bond with the substrate or the substrates. It forms a temporary bond. And when it forms that temporary bond, it usually is going to shift its position a little bit. And when the enzyme shifts its position, it puts some stress on the substrate molecule or molecules, and that is what speeds up the chemical reaction, okay? Now, when you start out with a substrate that's depicted here in orange, you will end up with the products, and here it's depicted as one big substrate getting turned into two smaller products, right? So you can imagine that over time, you're going to run out of substrate. Substrate gets used up in biochemical reactions. We run out of it. If you keep ripping it apart, you're gonna run out of it, right? This is important. Enzymes don't get used up. Enzymes don't. So the enzyme, it will grab a substrate, form a temporary bond, do what it does, let those things go back and do it again, and do it again, and do it again, and do it again. See, it is like a robot, right? Now, it's not an exciting robot like, does anyone know Rosie the robot from the Jetsons? Is it just me? Jetsons, best cartoon ever. Or like R2D2 or something like that. It's not an exciting robot like that. Enzymes are more like really boring assembly line robots that grab a bolt, put it in the car, grab a bolt, put it in the car. They're more like that. Okay? So substrate, products, enzymes. Enzymes are organic catalysts, almost always proteins. The arrow means the difference between the before and after. Okay, good. Yeah, assembly line robots. Good. Now, technically, this is how catalysts in general work. And remember that enzymes are organic catalysts. Catalysts because they make biochemical reactions happen faster. And organic catalysts because they're made out of a biomolecule and they're made by life, right? All of the different biochemical reactions that the enzymes that are in your body make happen, all of them could happen accidentally, but it just wouldn't happen very often at all. For example, inside of your cell, there are lots of amino acids. And every so often, amino acids will just bump into each other, and they'll just happen to be at the right position and the right speed that they just happen to hook themselves together. Okay? That could happen. But life would not be life if it waited for stuff to just sort of happen. Enzymes is our life's way of making things happen quickly enough so that we can be alive and making them happen where and when the living cell wants them to happen. And one of the ways it does this is by lowering the energy of activation for a reaction. It lowers the energy of activation. What's the energy of activation? Normally, there needs to be a certain amount of oomph to like put two things together or rip it apart. The amount of oomph that is needed for putting things together or ripping it apart is the activation energy. And an organic catalyst, an enzyme, makes the amount of oomph that's needed less. So technically, it lowers the energy of activation for a reaction. It's very often on an exam. Okay, let's see enzymes. Let's see if we've got a good video that you can see. Enzymes are. I don't think that you can hear this, but enzymes are proteins that speed up chemical reactions. See how we said enzymes are proteins? There's an exception to that rule, but yeah. The cell, a special. Now you can see the active site on this particular enzyme. And I really wish they would depict them as like little robots, but they just don't. So this shape makes it so that these molecules will bind in here. Maybe I should turn this down there, okay? So these substrates are going to bind in there. And when they do, there will be a temporary bond. Temporarily, this will be one molecule. Remember ionic bonds, right? And then the protein will change its shape, and changing its shape puts stresses on the substrates that it is in touch with, and that will be what lowers the energy of activation and makes this reaction happen more quickly. Okay, then the enzyme is going to release the substrate, the product, once it's done, and then it goes back to its normal shape and it's ready to grab more new substrates over and over and over and over again. How quickly an enzyme works has got an intrinsic rate. Let's watch it one more time. How quickly an enzyme works has got an intrinsic rate. Like every enzyme has got its normal speed limit. Like if you gave it all the perfect conditions, there's this maximum speed that it can work at. But at anything less than those perfect conditions, like not having very much substrate in the environment, for example, or having the wrong pH or the wrong temperature. Those are things that can slow an enzyme down. Now, in your lab about enzymes, actually, usually in your lab, we use hydrogen peroxide, and it's something that gets ripped apart. But right now, I don't remember what your online enzyme lab is. I shouldn't have said anything. Okay, so there you go. Enzymes. Enzymes make things happen. Okay, so enzymes make life possible. When enzymes put things together, putting things together is called anabolism, or is called an anabolic reaction, and ripping things apart is called catabolism, or a catabolic reaction. Anabolic, putting things together, taking two little things, boom, putting them together, like building a protein, taking individual amino acids, put them together, make a protein. Or we're seeing them here. This is an individual glucose molecule right here. There's a glucose, there's a glucose, there's a glucose, and an enzyme takes those individual guys and puts them together to make something like a starch or like glycogen. That would be an anabolic reaction. Now, catabolic reactions would be taking this starch, and like when you digest the starch that you've eaten, there are enzymes that cut it apart and release the individual pieces, and that would be called a catabolic reaction. Alrighty, there's probably more you need to know about enzymes for your lab, but that covers what we need to mostly know for a lecture. I will talk to you at the next topic, but we're done with DNA and enzymes for now.